Thank you. We are honored to be here. We'd like to thank the organizers for allowing us to participate in such an amazing evening. As she mentioned, we are both associate professors at Texas Biomedical Research Institute, whose mission is to eradicate infectious disease. And as part of that mission, we have a lot of really unique research resources. And one of those is that we're the only independent research center with a national primate research center. And I'm going to share with you why primates are so important for the work that we do, and show you lots of cute pictures because that's what I do. I don't study infectious disease specifically, but I study something that affects all of us that might make us at higher risk for infectious disease. So I like to start with the bad news, and the bad news is that we are all aging. Some of you better than me, as I stand up here with my walker. Hip replacement 2 weeks ago, so do a little dance. So, as we know, aging is a primary risk factor for a number of diseases: hip fracture, cancer, cardiovascular disease, Parkinson's disease, Alzheimer's disease. And these diseases have a massive impact on the individuals and make them at higher risk for loss of independence, loss of quality of life. And so my research is really focused on not extending longevity, but on focusing on that quality of life. Can we make things better longer? And we all know people that age really well, they look really great, they can have a hip fracture and bounce right back up and go run a marathon. Probably not in my future, but we can hope. And we know other individuals that age very poorly and are at higher risk for a cascade of diseases after the first impact and challenge. And so one of the reasons that we work with primates, like marmosets, which are small New World primates from Brazil, is to allow us to study this in a shorter-lived animal instead of spending our entire career studying one cohort of humans. And so marmosets are a great primate model for aging, and I've had the privilege of working with them since I was an undergrad. I always knew I wanted to be a primatologist. In grad school, if you'd asked me if I was going to study aging, I would have laughed at you because I was interested in big evolutionary ecological questions like parental behavior and male mate guarding and those types of things that probably reflected my life stage. I was having children and interested in those things. But when I finished grad school, I realized aging is a reality and became interested in aging and had the privilege of joining a very large consortium here in San Antonio that includes UT Health, A&M San Antonio, Trinity University, Texas Biomed, to study aging and to look at quality of life. And what we find with the marmosets is they age in a very similar way to humans. They have increased risk of cardiovascular disease as they get older. They have increased risk of dementia as they get older. And so we can start looking for the variability between individuals. So I'm going to show you a quick clip of monkeys doing a task that we designed for them. And this always reminds me of I Love Lucy in the Chocolate Factory. Right? So they love marshmallows. They don't like apples as much. So the task gets trickier, and they have to make a choice, and they have to control the impulse to wait for the marshmallow. Young animals learn this very quickly. Geriatric animals take a little bit longer, but eventually they all learn it. But we can see that variability between individuals. And looking at this variability in aging marmosets allows us to test interventions, which is what we're really interested in, right? We want interventions that allow higher quality of life. So Bill mentioned earlier rapamycin, there's metformin, acarbose, exercise. These are all interventions that might increase quality of life for people as we age, and we want to be able to test those to be able to recommend that to geriatricians so they can innovate new technologies and new things for the human aging population. And Dr. Daadi is going to introduce some of those innovative new techniques that they have.
Great. Thank you, Corey. Good evening. It's great to be in here. As a neuroscientist, one of my interest is anything that is research related to the brain fascinates me. And one of my expertise that I developed over the year is to understand the cellular and molecular mechanism that governs how a cell make a decision, how a brain cell make a decision to become any specific kind of brain cells. And these mechanisms are best studied using stem cells. And now with the cellular reprogramming, as we can reprogram any cell of our body, we can reprogram it to become a stem cell. This opens up a new avenues and new frontiers in regenerative biology and regenerative medicine. So my lab at Texas Biomedical Research Institute, we set up this discovery platform that it's utilizing adult stem cell reprogramming, and then and trying to discover some new therapies that is a novel approach to intractable diseases, basically. And this is through two ways, through drug discovery or through cell transplantation therapy, in which we can take a cell that is new, healthy, and young, we can transplant it back into an injured or diseased brain. And really, the goal of these studies is to develop a therapy that would stall or even reverse the aging, and also develop therapies for neurodegenerative disease such as Parkinson's disease, Alzheimer's disease, and related dementia. So what is cell reprogramming and how does it work? So if we take like a teaspoon of someone's blood, anyone in here in the room, or a skin biopsy for 1 millimeter, we can take those cells, we can grow them in a dish, and then we can reprogram them. We can insert 4 genes, and then turn the clock back of this cell. Basically, took the aging clock back and push them into a stem cell status. And this stem cell state is called pluripotent stem cell. Once they reach into the pluripotent stem cell, they can give a rise to any cell of the body, and that's what those 3 colonies that you see on the back there. We can generate those, and we can generate billions of them. So we can see the potential of this approach to be able to generate various kinds of cells. So my expertise is really on brain cells. So we can take these cells, we can convert them into a brain cell, which is, this is an organoid. We can also call it mini brain. We call it mini brain because it has all the principal cell type of a brain, which is neuron, astrocytes, and oligodendrocytes. So we can imagine, so this is a biopsy that is taken from a patient and represents really the brain of that patient. So this is within the realm of really patient-centered medicine. But in addition to that, we took a cell and we kind of coax them to become heart cell. And in this situation, the cell self-assembled and made a beating heart. So this is one of the best assays that you can test a drug, for example, for heart conditions. But my expertise is really on Parkinson's disease. And Parkinson's disease is caused by the death of dopamine neuron in the midbrain. So when the cell, they die in the midbrain, the communication with the forebrain is interrupted, as you see in the tweeting arrow in the blue. When this communication is interrupted, the patient starts to develop these symptoms, which is tremors and slowness of the movement and rigidity. But if you take a look at the cells inside the midbrain, you took a look at them a little bit more carefully, you can see that the cells, they die because of this protein inside them that gets accumulated. When it gets accumulated, it kills the cell. This protein called alpha-synuclein. The problem with this alpha-synuclein is also propagates inside the brain. It jumps from one cell to another cell, which is an amazing problem that we have now we have to address. So it's in a prion-like mechanism. So this is something that we need to attack, and what we did is we went out model it in vitro. So to model it in vitro, we created a mini brain, which is, again, it's a midbrain organoid that is from the midbrain. We have the dopaminergic neuron, and those are created from a skin biopsy from a patient. So again, it's really patient-centered, and we took also some tissue from a brain that have Parkinson's disease. We isolated alpha-synuclein from that patient, and then incubated those organoids with alpha-synuclein. And then we have actually now those alpha-synuclein infected the brain organoid, and in the meantime, we have now a mini brain that we can, exactly like a Parkinson's disease in a dish. So this is one of the best assays if you want to screen for a drug, for drug screening. You can really scale it up, and as you can see in here, those are little organs in there. Those are little mini brains. We can grow literally billions of them. We can produce them in a mass and also preserve them, and we can plug them into a high-throughput screening drug discovery effort. And in the other approach that I mentioned to you, which is cell transplantation, we can take a skin biopsy, and you can see those beautiful neurons on the left side. Those are dopamine neurons. They secrete dopamine, and they are derived from the skin. So because I mentioned to you the interruption between the midbrain and the forebrain is lacking dopamine. So the idea is to inject dopamine cell back into the forebrain and reconstitute that area. So and this is something that it's been actually developed already published and been tested in humans as well as in preclinical. But they were using other kind of cells that are a little bit controversial as well as unreliable. But now we can derive them from a patient's own skin. So this is really very promising. But before moving there, what we did is we developed an MRI-guided injection to be able to deliver these cells safely inside the brain. And this was done in collaboration with the UT Health team in the Research Imaging Institute. So we developed this therapy in which it enables you to deliver the cell exactly into the area that you want under MRI. This is real time, and enable you also not to make any potential mistakes, for example, hitting a blood vessel or hitting a structure that you don't want to. So this is real time within the OR, and you take in the image, you collect them from the MRI, they come in, you put them in a 3D, and you enable you to touch. All you need to do, the neurosurgeon, is to touch the spot where the cells is going to end up, which is the forebrain, areas that are lacking dopamine, and put the cells right there. So this is quite, and this is how we envision this therapy to be in the clinic, to enable the neurosurgeon to access the area very safely and deposit the cell where it's supposed to be, and that's to increase also the efficacy of the cells. But really, why we do this, it's really it's for the patient to really increase the safety and also the efficacy of the therapy of the patient. I have the privilege to meet quite a few patients in San Antonio, and it's been really a privilege to have the support from San Antonio to Texas Biomed, and when you see them in their eyes, you can see the situation that they are in. And I can tell you, with this therapy, it's going to transform the way we manage Parkinson's disease. Thank you.